quality of the produce and surety of the farmer's income, and also surety of the total produce needed by the population in the world. Moving forward, let me go through that. This is a basic question. Everybody is talking about the population which is we are going to have. At present is close to 7, and it is cross 9 billion people. Now, 9 billion people with present production around 2.5, 2.6 billion tons of food. We need more than 3,200 million tons of food. So the food requirement will increase, and by any mean, by another 10 to 15 years, the 50% more food is required, which we are present producing today. So if you see the demand in the developing world is higher, and overall world will have higher requirement of food. And if you see the yields are also now increasing, and but in a few cases like wheat, soya beans, they are now not much increasing in unless there is a new technologies are required. But maize is where a lot of exploitation is happening, and particularly in the developed worlds like US, that the yields are increasing. But in the tropical world, where the growing seasons are lesser, the yields are not growing much, and particularly in the Kharif season, where the rainy season, where the temperatures are high. So, what is now needed for the world, and particularly for the developing world? And developing worlds we talk about Asia and Africa. Now, here the question is that what should be our approach? Because what is our resources? And these resources are not plenty now. It is decreasing. The land resource is shrinking because of urbanization, degradation, the fertility levels, the soil health. Everything is there. Everybody is talking about these days about these conditions, and above all the climate. And the climate change, the shifts of climates, that we are not getting the rain as per the rainy seasons. Either we're getting early, or we're getting late, or we are getting plenty, or we are getting deficient. So there are a lot of things are happening. The temperatures are increasing in some places, and some places the temperatures are going down. So all these parameters are demanding a new crop types, the new plant types, which can withstand these kind of stresses. Beside that, with the change in climate, the diseases, the pests also changing. So we are dealing with many challenges which we have in crops. So what are the alternative options we have? So our options are that unless we use something different, because we call about our variability, we call biodiversity, which is also a talk of the word now about biodiversities, and we talk about CBD laws, we talk about the gene pool exchange, we talk about the biodiversity acts of the countries. So everything is a challenge. It is not free now that you can get germplasm. But we have seen in our times, when we were doing our PhDs in 70s, 80s, or 90s, the germplasms are available on call, and you just write to the people, and they will send you the germplasm. But after the CBD laws come into the force, everybody is talking about the country's heritage. And then there is a kind of system is coming that unless you pay for that, or unless you create a remunerative system to the countries, the germplasm will not be exchanged. So then the CBD, then the Food and Agriculture Organization, which is the key organization to ensure food supply in the world, found out that how we can segregate the agri-biodiversity from the general biodiversity. And agri-biodiversity, when it was talked about, agri-biodiversity means that you need germplasm, which is for the food crops, food crops include vegetables, fruit crops, and also the cereals and grain crops. So they created another treaty for the world, which is under the aegis of FAO and also CBD, is called International Treaty on Plant Genetic Resources for Food and Agriculture. Now, this treaty ensures that the germplasm exchange should happen through the multilateral system. So multilateral system means that all signing countries, almost 100, 600, 7 countries have signed this treaty. And they say that under this signature, they will share the germplasm with the gene banks through the multilateral system. And each user will get through the standard material transfer agreements. It is called SMTA or MTA. So these are standard procedures where they will apply and get this gene pool. In turn, they pay around 0.1%, 0.01% of their turnover when they develop the crop through this germplasm back to the gene bank or to the treaty. And treaty will then offer this money to the countries for conservation and protection and development of the gene pools. So this is what the procedure is. Now, this procedure is becoming more tougher for the users, and clarity is still not there. Many countries are not sharing their germplasm under multilateral systems. Everybody is looking into the local gene pools and protected gene pools, and they want to get the benefit within their country, and they want more share of the benefit countries which are rich in gene pool. So I'm explaining you what are the challenges which are coming in developing the food crops or doing breeding and developing new seeds which can yield most. So then a new treaty under treaty came that Nagoya Protocol. Nagoya Protocol in Nagoya in Japan, every country decided that they will pay at the time of development the product a certain amount of the turnover to the central system, and that money will be used for the conservation. But then the seed industry, which is the major user, and both public and private, they decided that no, this will not serve the purpose of the treaty, because the money will come after 10 years. Because if you take germplasm today and you will start developing your variety or a hybrid, it will take 7-8 years, and then you try to test it. So minimum 10 years it will take to develop a hybrid, and then you start giving the money. So we decided as industry that we should give another model for giving that money to the treaty more upright. So we proposed a model called subscription model, that every year we will pay part of our turnover, a small percentage of turnover by the user to the treaty, so that the minimum corpus of money should be created for the world to have the this germplasm, and every industry should get germplasm as and when they require. But this system is still under negotiation, and the last meeting happened in India, and GB9 meeting, and we all participated. We tried to convince the countries, but each country has to take their call. So it is maybe inching towards that. The second issue was the crops, which are included in the Annexure 1 of this treaty, which are under MLS. So everybody wants that important crops are missing. So we want to expand this Annex 1 treaty document. And this also was discussed, and a special group was created. And this group will present the report in the GB10 or GB11. So what I'm saying is that the challenges are there. So, but these challenges have to be overcome for the breeders and the geneticists and the companies who are breeding for such crops, so that they can give a better product for future higher production per unit area, because area we cannot expand. Water shortages are there in many areas, and the other constraint of biotics diseases are there. You have to overcome these things. So the challenge for the breeders are many, and challenge for the planners are many. The challenge for countries are many, but food production, you cannot take a risk. You need food. You need good quality food. You need good food for your whole bulging populations. Otherwise, you will face trouble, the wars, the uneasiness within the populations, and this is happening in the world. So I think what is required is that the surety of food, and that is what the mandate of the United Nations, that the food should be the first thing we should protect, provide, and give nutrition to the populations. So I think this is where situation is now moving, and but the technology which will save our life. Before we talk the word of digitalization, we talk about many things. So if you see here the same thing which I was telling you, that what is happening, that the plant breeders encounter that you have to stabilize and increase yield despite climates, because you have this United Nations. You might have seen the SDGs, the Sustainable Development Goals, which are 17 in number, and out of that, which relevant to the agriculture is around 8. And you can see that there is the poverty issue, the water, the nutrition, the food. So I think what I'm saying is that you have to have maximize resource use efficiency, whether water or land or nutrients, because these are the limiting factor for the agriculture. Well, if you see the climate change, the temperatures, they say by in 2016, 14 degree to 16 degrees, so 2-degree temperature, global temperature will increase, which will put pressure on the agriculture. The pests, because of the climate, they become more robust, or they also mutate, they also develop. So it will challenge that how to handle them with the new technologies, or the new spray chemicals, or with genetics. That is a challenge. The water is a challenge, availability of water. Population is a challenge, arable land reduction is a challenge. So if you see that these are challenges, but we are not, I'm highlighting only the challenge that it can't be handled. But what is the response of the breeders? How breeders are going to respond to the challenge? So I think that is what we, as an International Seed Federation, where I am also part of that committee of plant breeding technologies. So the innovations which we are doing, that will bring some solution to all these stresses. And I think this is all we are working towards that, and this is what our goal is. Now, if you see that the whole process of technology development in this agriculture sector, see, we say that when the early farmers, they just making selections, they are going to the areas where species are growing. They start selecting a good plant with better looking seed, or the spike where seeds are there. So it starts selection. So is long history of 10,000 BC, when the emmer wheat or small wheats, which you might have seen the different species of wheat. Then the people start selecting from the maize initial varieties, or the species, which you can even cannot even identify that it's a maize. So people have done it. So the selection process started long back, and then slowly the selection come into the we make it crosses, the Mendel laws. The Mendel laws came in 1866, when the Mendel law of genetics started, the law of segregation, law of independent assortments. And then we see that how the genetics works. We say that during, when the gametes are formation, there is a meiosis, and the segregation happen, and then the recombination happening during this, and then you get the new recombinants. So we start making crosses and start making the selections, and in the early phases. Then people have used the cereal, cereals, and other to make a triticale, which is like we call man-made cereal, because this cross doesn't occur in the nature. So they created, and that bring another kind of revolution in breeding, that how they can do it. And then we started the hybrid breeding. We see that there is a heterosis, that if you cross diverse parents, you get in the first filial generation the advantage of that, and your yields are higher. And that is how the heterotic group or the heterotic population, when they cross, the heterosis bring better yield. And this is what presently most of the seed companies, if you see, they are using this technique, that how to use these hybrid breeding technique. So they use this technique, and then start developing the new types, the new recombinants, the new varieties, new hybrids, which yields higher than that. And if you see today, maize is a good example, where the yield, which was before that around 1 ton per hectare, now reaching to almost 11 to 12 ton per hectare, and in future maybe lead to 15 to 16 ton per hectare, which is a great improvement of the yield per unit area. And then they came with the mutation breeding, and people say that with the mutants, chemical mutants or radiation mutants, people have used that technology to mutate the genes. There is a natural mutation was also happening because of the pollutions, or because of the radiations, because of the other things. Natural mutation means the natural development also is happening in the species, which we sometimes we call it land races, where the genes start mutating, and because of the environmental pressure, we call it G into E, the genetic into environment, and that bring the new types which are more adaptable to the situation which they are growing. And this adoption takes longer time, but in nature. But then scientists, they say that why not to do in the laboratory and field itself, to bring more mutation, targeted mutations. And then you do something like you can delete a certain portion of the genome, and get the alter function of the genes. And that is what you are doing. And then people use the tissue culture, and tissue culture also bring another advantage of that you are capturing the somoclonal variations in the tissue cultures, and you are finding better types through tissue culture. And also then people started using the haploid culture, the make it double haploids, where homozygosity is showed 100%, because we are using microspores, or we are using the tapetum cells, where the meiosis is happening, and then we diploidize it. So I think these technology help breeders to develop the new types. And then moving forward, then when the in early 80s, when the gene and DNA sequencing happened, people talk about that how we can use this new technology in this. So what we can do is that then we have started looking into the genome itself, because there is a lot of new machines developed which can PCRs and other things, where we can see the genome very clearly, and try to understand the gene functions by seeing the expression and other genes. And then we start developing a new technique which is called the genetic modification, because there's a lot of development of technique developed to insert the gene in the genome itself. So people have identified a few we call it interspecies transfer, not intraspecies, but interspecies transfer. And they transfer the genes from the other species from into the higher plants, from bacteria, from non-crossable species, and try to build the new traits there. And the first example you might have seen, the cotton, BT cotton, the gene came from the bacteria, the soil bacteria, Bacillus thuringiensis, and this bacteria has a protein which act on the gut of the insect and just kill the insect. So this BT, because BT sporulation, the spores were initially used as the pesticide sprays, and based on that, the people transferred the gene rather than spray. They put the gene inside the plant, and the expression has happened, and so it controlled many lepidopteran insects. So this is a big revolution. And also in maize, the same revolution happened. And then people find out another gene, which is a herbicide resistant gene, and they try to put in many crops like soybeans and other crops, where they find the plant itself carries the gene, so there's no need of using external thing to control, but you can use the herbicide, and it will select it will kill only the weeds, not the plant, because plant has a protection with the gene. So similarly, I think these technology help us to safeguard the yields of the farmers, because we supply them a new genetics, the new seed, which help them not to miss the control, because they are doing it with the help of initially chemicals. A lot of chemical sprays were happening, but now they are totally immune to bringing that gene inside the plant and then do it. And same time, there is a people start using the selection markers, because now the genome and the gene functions were known. So people start developing the markers, and then when they breed it, when they make populations, they can just pick up the plants which have the genes. And so that being a smart breeding, that the breeding come very short. So they start making a lot of these new markers, identify the markers in the populations by creating mapping populations. So first there's the AFLPs, new technology, then developed AFLP markers, then came to the SSRs, and then slowly now this is SNPs, the single nucleic polymorphism has been identified. So the technology is developing, and similarly these technology helping breeders become more accurate, precise, and also do the fast breeding, because he ensures that the genes are there. Though he has to do the breeding in the environment, because environment factor is the expression, but a lot of time he saved, and a lot of space he is saving, because he identify those plants where the genes are there. So he that, and then become the whole genome sequencing, and people go the genomic breeding through the whole genome, the sequencing and haplotyping, so they exactly know where the alleles are there, where the genes are there, and then they try to with the help of software, they try to build a combinations which carry those traits, so that you know exactly where your breeding is heading towards. It's not hit and trial. It is not that you make a lot of crosses and make a selection. And then there is a technology come the gene editing, which is the further precise, because the reason it was found that the virus bacteria interaction, that when the bacteriophage, they are affecting the bacteria has a system which can understand that which virus is coming, so it can cleave the that and save itself. So the very precise cleaving. So people develop the CRISPR-Cas9 technologies, which are palindromic regions, repeat regions, which people found. So with that, the new system has come. Now people are, if you know the sequence, you know the gene, you can mask the gene function, you can move the gene function, or you can improve the gene function. So I think these technologies are heading towards such a way that it brings a lot of precision, a lot of scope, a lot of opportunity to create new traits, and traits which are required for biotic and abiotic stresses. More and more information is being generated, but one caution word: we can't beat the nature. Why? Because the genome is functioning as a whole. It's not working tandem. It's working as a whole. So you have to understand I call it three environmental system: the nuclear environment, the cytoplasmic environment, and the external environment. So these three environments are interacting, and there are a lot of signals. A lot of signals are going inside, and a lot of signals are reacted by the cytoplasmic activities and by the nuclear activities, and they whole decide what is the functionality of the whole system. So I think these are some of the things we have to keep in mind, that how the development has happened and how many years it takes, but it is going faster and faster. And now people have the new tools. You feed the data, you can understand which which inbred line I cross with which inbred line, and what the outcome, how much heterosis I will get. People are trying to establish these new algorithms with the new databases, and I think it is going to be a very advanced and complex systems which people will use it to give a better product outcome to the breeders. So I think if you see that in the as I told you in the tropics, we need all these innovations, genetic innovation. Without that, it's difficult to work, because tropics are the most variable environment which is challenged the breeders more, because the climates, the temperatures, these systems are there. Also, we need strong seed system, because not only we do the innovation, but this innovation should be transformed into a commercial product, which can go to the market. And commercial product, if you go to the market, need a good seed system. So we have to develop a good seed system, and along with it, the good agronomy, like I told you, the herbicide resistance, the other resistances, so that there should be a selection, a smart selection by a particular chemical or by any technology, that it distinct between good and bad. So I think this is what the combination of solution we have, along with the enabling policies. We required a crop yield for better for the livelihood of the smallholder farmers. It's big talk these days about smallholder farmers, because farmers are becoming small, and their livelihood is based on this. India has a huge population of farming community. We have around I think 1,400 million, 140 million farming families who are engaged directly. So I think, and if you see in the population, almost around 58% population directly or indirectly involved. So we have to protect them. So I think this is what our job is, that if farmer is not getting good yield and good remuneration, he will be out of the agri business and shift to the other businesses. And that is what is happening in very well-developed countries. And this will start happening in better remuneration. So I think unless we find a good profit and benefit in the farming system, it's difficult. So the plant breeding, as I told you, it takes longer years for develop new technologies to come into the commercial side. So you see that how much efforts and time will go into that. And this will bring another facets, that any technology which we say, it is not easy. It is very easy to say that now we can do anything. But when we stabilize and bring a product, it takes another long time to bring it to a level of cultivation where the benefit go to the farmers and the populations. So I think this whole thing is if you see the timelines, a huge number of years have been there, which has been spent long back, and bring to the level where we are today. And we are now running towards more precise and good breeding. So the breeding case, which what we call it, breeding equation, that we need the phenotyping, which is important, because any trait which we are putting it, we need to see that whether it is expressed there or not. And now new technologies are coming for phenotyping. There is a digital phenotyping, and you can phenotype many traits together. So your selection intensity will increase. Your field variability, particularly the selection accuracy, will increase. Your time will off season, or see, like I told you, double haploidy system, that will reduce, and then you get the genetic variation which is in the gene banks. You can, if you are phenotyping rightly, put in the annotation systems which can give you the variation which can be used properly in making the new crop system. So I think the breeder has a challenge. Breeder use the new innovations, has to use the new innovations, because otherwise the time is not there. The time is the changes which happened previously in 100 years or 200 years now happening in say in few decades. The time is not with us, and this variation is increasing. because the population which takes 1 billion in initially 150 years, 160 years, now 1 billion increase of population is happening in 1 or 2 decades. So you see that, and the changes in climate, the changes in temperatures is happening very fast. And a genetic variation and breeder has to really on the toes to develop product which can suitable to these changes. And these changes are really happening very fast. So I think this is what message I can give it to you is that the challenges are many, but there are chances which every geneticist is using, that how we can make do the smart, precise, and speed breeding. Well, speed is now everywhere. Everybody talk about these days the speed. If you see, you want you just phone to Zomato and you want the food within 15 minutes, and or you go there, you want to reach to a place within an hour, and you want to travel internationally within a short time. So I think everything is a time. So similarly, this breeding, they also want the speed in with which they can bring the new products and better products. So this is what I want to say, and I think the things are open to questions if you have. Thank you.
Thank you, Dr. Arvind, for a detailed presentation. I'll take up some questions. So, first question, what are some new breeding technologies being used in the development of hybrids and varietal seeds?
See, as I told you before, that now we are using the gene editing, we are using the sequencing, we are using markers. So we are using phenotyping. We are using we call it speed breeding by providing 18 hours light and the growth conditions. So there are many technology which we are incorporating now. Yes, there is a expensive tool. Some are the expensive tools, but everybody is looking into the speed of getting the products. So we have to use these, and I think we are using now most of the geneticists, whether in our country or other countries, start using some of them, and ultimately everybody will use everything which is available. So there are technologies available. Gene editing, I told you one. Marker selection is a little bit before. We are still using marker analysis. We stood also gene sequencing and expression EST tags so that we can see the expression of genes. So phenotyping, we use the digital phenotyping, and we are using now different IT tools, systems we have created by putting data and annotating data towards seeing that which of the material we cross with what to get a best and better yield. The trait can be recombined in such a way that we get a better output.
Okay. Next question, how do these new breeding technologies contribute to the improvement of crop yields and quality?
So this is what is the final goal. The final goal is that you have to get the best recombinants. So the final goal is always in mind, because these technologies are expensive. We don't use for fun. We use for creating the products, the commercial products, the better yield. I told you that in one of the program of Corteva in US, they have presently the hybrid which can yield 12 ton per hectare. And now they are working another 5 years. They want to reach to the 15 ton per hectare. This is what the focus is. So this focus will come from where? They are creating heterotic pools in such a way that all the traits are accumulated in inbuilt lines. Then using the system, IT system, they try to work out that which genetic variation cross with which genetic variation to get 15 ton yield. So I think this is what these technology will do. And the traits which are difficult to get in the natural populations, we can silence certain genes. If we know the gene function, we can enhance the gene function. We can see that those we can switch on and switch off certain genes in such a way and modify them in such a way that we get the better output. So people are trying. The technologies are being perfected now, and yes, you will get the product which have better traits, better stress tolerant traits, better pest and disease resistant traits, and better yield traits. because yields are there, but it is eaten away by certain stress factors which has to be avoided so that you can maintain the genetic potential of the crop. This is what we are doing.
Okay. And what are the advantages of using hybrid seeds compared to traditional open pollinated varieties?
See, the traditional open varieties have their limitation of the yields. What heterosis does is that we know we cross two diverse parents and bring recombinant in such a way that the first filial generation, which is a known fact of heterosis, is that the F1, the first filial generation, has the better performance than both the parents. So that is what heterosis means. So I think hybrids always have the advantage over the varieties because of their higher yield, better combinations, and better expression of certain traits, which a variety, because variety is, we call it varietal improvement as more towards homozygosity, and they are more stable so that they bring certain traits there. I'm not saying their approaches are not. Certain crops we can't make hybrids and we are only making the varieties. But varieties are equally important. But where hybrids are possible, this technology is bringing more improvement in the yield and more improvement in the trait expression.
Okay. And how do new breeding technologies ensure the stability and uniformity of hybrid and varietal seeds?
See, this is what we are doing. We are using technology, and it is actually a combination of new technology and conventional breeding. Both are required because conventional breeding is we are trying in the actual fields where the crop is growing. So we are taking into account all the conditions there, whether the environmental conditions, soil conditions, cultural practices, and we are growing within with the new genetics in these areas, and then we ensure that they are stable under these growing conditions, and that is what our goal is. So we do a lot of trialing in the fields and going initial material, selecting there, going into the hotspot areas where diseases are prevalent, and checking the stability there, and then bringing to the level of hybridization or new varieties, which are again tested, again going back to those whether those genes are still there or expression is still there, and expressing in a right way and giving you the same output which we want from the gene to be there. So that is what our goal is, to stabilize, and that is how these new hybrids have a stable performance, and they tested for stable performance. That is why farmers are buying seed every year and getting better yield.
Okay. And how do new breeding technologies ensure the stability and uniformity of hybrid and varietable seeds?
Same thing I explained now, that how it is ensuring, because that is what our goal is, because we know exactly what we are doing.
Sorry, yeah, correct. Yeah. And are there any potential challenges or limitations associated with the use of new breeding technologies in seed development?
Yes, there are policy issues are there. People talk about, because we have a lot of people who say that we are doing something which is not in the nature. But I think we have overcome many of the challenges. We are testing that we are not bringing any kind of anti-nutritional factors or anything which is not good for the human being or animals. So I think that is our procedures. Our testing procedures are very stringent, and we are going through all the tests before we release the product. So there are enough controls and policies available, and new systems are now being developed. New testing systems are coming. So improvement in everything is happening, and I don't think anything bad will be released in the environment by any geneticist. Yeah.
Yeah. How do these technologies impact the time and cost required for development of new hybrids and varietal seeds?
Well, time will be reduced, but cost will increase surely. And that is what our goal is now, how to, because there is a benefit, because if you are getting advantage of the time and the accuracy and the speed, you have to pay higher cost, no doubt about it, because these things are expensive and technologies are expensive. But in the end, we want to get food availability for the populations. I think it is worth taking the cost, because at least we are spending on the food availability, and many countries are spending money on the armament and other things, which is counterproductive for the population. But at least we are spending money for the food, and it's worth spending money, which will give you better food and plenty of food in future.
Okay. And what are the potential benefits of using varietal seeds developed through new breeding technologies for specific environmental conditions or regions?
See, the problem is that I told you, the climate change. The problem is that there are a lot of variations are coming. So to make it possible, those like biotic and abiotic stress tolerant or resistant, you have to create those kind of genetics, because even focus is that we should not spray chemicals which can save environment of pollution. So we want to bring genetics in such a way that the most of the traits should be controlled by genetic itself, rather than by external spraying factor. So our goal is very clear. It will take time. We are moving towards that, and with the new technologies. And like in BT cotton, you see that how much sprays are saved, and farmers' health has improved, soil health has improved, environmental health has improved, because the lot of sprays are avoided. So I think with the other technology also, we'll try to reduce dependency on the chemicals, and try to develop cultivars which have better resistant to the diseases and tolerant to the temperature variations, drought conditions. With less water, we can grow crops. With high temperature, we can grow better yield crops. Again, with the low temperature also. So there are we are developing those kind of traits which can help us.
How do new breeding technologies address the demand for crops with improved resistance to pest, disease, and environmental stresses?
Yeah, the same thing, because we are now understanding the genome well. We are understanding where the which genes are involved for the susceptibility, which are gene for the resistance. So based on that, we are using that. I told you that with the gene editing, we can edit that gene, and accordingly, so that we can get the right expression, and those susceptibility should go, and resistance should come. So that's what our goal is. So that is how it can handle the biotic and abiotic things.
Okay. There's a couple of more questions. What are the ethical and regulatory considerations associated with the development and commercialization of hybrids and varietal seeds using new breeding technologies?
See, the GM debate has quite a bit intense before, but now slowly dying down, because people have understood that it is well-tested products, and there's no damage happened to the environment or to the human being or to the animal. So ethically, we are bound to do that kind of things, and we are very open to the scrutiny, and we are doing quite a bit trialing of human health checking or environmental health checking, and we do a lot of analysis, chemical analysis, so that we should not release any product which has higher of any toxicity or any damage to the environment. So we are very careful about it, and now it is rather more accurate than previously. When we were doing conventional breeding, it was never thought about that, but now it is very clear that we, by using technologies, we exactly know what we are doing, and it can be tested, it can be measured, it can be seen. So I think we are conscious to these issues of ethics and policies.
Okay. So finally, we have the last question. Please let us know a little about your career background and how you got interested in this field.
Well, I have now spent 43 years in agriculture after my PhD. I did my PhD in 1980 in molecular genetics. long back, and after that I worked in the plant breeding department of PAU for 8 years before moving out into the private sector. And my career in private sector is 35, 36 years now. And I did a lot of participation in the breeding, and worked in not only in India and also for Asia and China. And we find out that how these things, the germplasm, the variation, the diversity is happening. Now I am chairman of the IP committee of Asia Pacific Seed Association, looking into the biodiversity issues, looking into the plant protection issues, looking into the trade issue, looking into the new technologies. I'm also part of the IP committee of International Seed Federation, where we are looking into the new issues of breeders' rights and farmers' rights, and how they are being handled in the world, how a breeder can be remunerated, and how farmer can get benefit. All these things I'm part of it. So my career and my long career and my interests are many. Yeah.
Okay. Okay, Dr. Arvind, I don't see any more questions from our participants. So now we have come to end of question round. On behalf of agricultureinformation.com, we like to thank you for a detailed presentation and answering all the questions in depth. And we also like to thank all the participants for joining this meeting. The meeting will now be closed. Have a good and safe weekend.
Thank you. Thank you a lot. Yeah.
You're most welcome, sir. Thank you.
Thank you.